So I'm really, really, really honored and actually humbled to have Dr. Eric Green as our keynote speaker today. Uh, Actually, I was thinking it may be better to have Dr. Parker, who is a professor at U of M, and then maybe introduce Dr. Green, which will be a much better way. So, I will go to.
Yeah. Thanks. Hey, Eric, can you hear me?
I can, Stephen. How are you?
Wonderful. I'm doing great.
Good.
First off, let me say congratulations to everyone in the room here. This is some amazing science that I've seen today. You all are so impressive. And what a treat you are in for today. It's my privilege to introduce our keynote speaker, Dr. Eric Green, who I'm fortunate to have crossed paths with during multiple stages of my own career. He's one of the smartest, kindest, reasonable, fair, knowledgeable, insightful, and overall amazing scientists and humans that I've ever met. We're very fortunate to have him today as a keynote speaker. Dr. Green earned his MD and PhD degrees in 1987 from WashU in St. Louis. Coincidentally, the word genomics was coined that same year, almost as if it was destiny for his career. And he's had quite a prolific career. He has authored or co-authored nearly 400 scientific publications. Dr. Green has earned multiple honors and awards, including election to the National Academy of Medicine in 2023. Dr. Eric Green spent most of his career as a genomics researcher, Human Genome Project participant, and former director of the National Human Genome Research Institute, or NHGRI, at the U.S. National Institutes of Health, or NIH. He was the third NHGRI director, having been appointed by NIH Director Francis Collins in 2009. He served as NHGRI director from 2009 until 2025. I did my postdoctoral fellowship training at NHGRI from 2009 to 2014, so I was a direct beneficiary of Dr. Green's leadership during those years. He created an unrivaled scientific research culture at NHGRI, one of openness and marked by world-class scientific rigor that was even fun. He had the vision to foster and support the very technologies that enable the bold genome science that happens today, including much of the work that's been presented here by all of you. Thank you for that, Eric. Dr. Green was at NHGRI for over 30 years, during which he was appointed to multiple key leadership roles prior to becoming director. He served as the Institute's scientific director for 7 years, chief of the Genome Technology Branch for 13 years, and founding director of the NIH Intramural Sequencing Center for 12 years. He knows a thing or two about genomics. For just over 2 decades, Dr. Green directed an independent research program that included integral start-to-finish roles in the Human Genome Project and groundbreaking work on mapping, sequencing, and characterizing genomes. Dr. Green, thank you for your leadership, insight, support, and kindness that led to such a strong genomics research foundation in this country and that has benefited not only the young researchers we are seeing here today, but also led to many medical breakthroughs that broadly help human society. So please join me in welcoming Dr. Green today.
Well, thank you. Pleasure. I had no idea I was going to hear from you today. I had no idea you were introducing me. That was a pleasant surprise for those who may not appreciate it. He seems a terrific trainee, now faculty member, University of Michigan. We are so proud of him and what he is accomplishing. He's emblematic of the kind of excellent science I'm going to tell you about. So I'm delighted to join you today. I always say yes when asked to do things like this. And what I thought I would do, recognizing that that many of you know bits and pieces about genomics, but I thought I would actually tell you the story of the field, really from the beginning, and really make this a story. So a lot of this is going to be history. Some of it is going to be current science and medicine. And at the end, I'm going to even take a little bit of a peek into the future. But I like talking about this story. That's why it's sort of my version of the story, because I think fundamentally it is so important for everyone to be grounded in some fundamentals of genomics. So, I mean, why should you care about this field of genomics? Obviously, I'm biased, but I think genomics is scientifically just way cool. It is associated with some remarkable history of exemplary scientific achievements that I think are absolutely worthy of mentioning. It is also is becoming increasingly relevant in everyday life. One of many examples of this is it's now relevant to human health and the practice of medicine. And so be prepared to be convinced by the end of my presentation that you should care about genomics. It is fun, it is cool, and it's important in your life. So what's the fundamental question that sort of led to the field of genomics in many ways? And there are a lot of questions you could ask, but one of the most fundamental questions humans have been asking is, what is the blueprint that is used to build and operate one of these, a human being? What is that fundamental blueprint that is used to build and operate us? Well, I can tell you that two scientific fields that were launched last century really brought great clarity to that question. And I think it's important to realize that they really were two different scientific fields with two words that were coined about 80 years apart. First, there was genetics. And genetics was, the word was coined, the field was launched at the beginning of last century. And genetics is the study of heredity. It's a study of how is it that traits are passed on from one generation to another, from one cell to another. There was great interest in untangling inheritance in the early part of the 1900s. And eventually, they figured out that DNA was the molecule that transmitted information about heredity. And at about the halftime or halfway point last century was when DNA's famous double helical structure was discovered. That was about 1953. But then what transpired once having knowledge of DNA structure was an accelerating pace of understanding how to study DNA, to the point where it almost became clear that we have the tools in hand to be able to be very comprehensive in studying all the DNA of an organism, and all the DNA of an organism is a genome. And that gave rise to this new word of genomics. And genomics really is the study of all the DNA of an organism, its genome. And that happened in 1987, as you heard, actually the year I graduated with my medical and graduate degrees. But it was 80 years after the introduction of the word and the launching of the field genetics. Well, to fully appreciate how it is that the genome really is that blueprint material for making and operating a human, let me just give you a little bit more biology. I bet most of you know this. The human body, for example, has something on the order of 30 to 40,000, or 30 to 40 trillion cells that make up the human body. Every one of those cells has in the middle of it a structure called the nucleus that house these things called chromosomes. Chromosomes are fundamentally the suitcases that carry the DNA from one cell to the next, because that's what chromosomes largely are responsible for, is carrying and packaging up all of this DNA. Now, it turns out that the human genome consists of about 46 of these chromosomes. They come in pairs. You get one set of 23 from mom, one set of 23 from dad. And in total, if you take all the DNA from all of those chromosomes, one representation of 23 pairs, and you really look at what it's made up of, it's made up of a series of building blocks, chemical building blocks, that we abbreviate G, A, T, and C. They actually have chemical names. We don't care about the chemical names for this. It is the building blocks of DNA are just made of 4 chemicals. But notice there's a very precise order on each of the strands of DNA, and that's called DNA sequence. And the order of the DNA letters, the G's, A's, T's, and C, have embedded within a code that is necessary for creating and operating a living system like a human being. If we took one set of those 23 chromosomes, like one set, the one you got from mom, the one you got from dad, and we added up all the letters, one representation of the human genome is 3 billion letters, or 3 billion bases. And so fundamentally, one way to think about this is the answer to the question, what is the blueprint used to build and operate one of these? It's basically 3 billion letters of code that can be represented just like that. G, A, T, C, 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 A, G, A, T, A, C, and on and on and on and on across 3 billion letters. That is fundamentally the blueprint for making and operating any living system, including a human. Well, what happened, and I alluded to this, is that starting with the discovery of the double helical structure, more and more knowledge came out about how it was that DNA was the information molecule. Some of you might have learned in school already about the genetic code. That's that famous lookup table that was discovered in the 1960s that tells you how it is that you can go from DNA sequence to actually encoding information for building proteins. Proteins are made up of amino acids. They're sort of the bricks and mortars of cells. And that was discovered in the 60s. By the 1970s, we figured out actually how to read out DNA by doing DNA sequencing so that we could determine the letters, the exact order of the letters in DNA, because that's what encodes the information. And then by the 1980s, there became all sorts of refined methods for DNA manipulation, be able to clone DNA, to be able to amplify DNA, to be able to manipulate DNA, all in the laboratory. And so this accelerated progress throughout the 1980s gave momentum to this idea of could we put these tools together and eventually figure out how to map and sequence all 3 billion letters of the human genome. And that is why the word genomics was coined in 1987, as I told you about earlier, because it was recognized that there was this exciting new discipline that needed a new word, it needed a new name, and it even had a new journal. And the reason a field was named and the reason why the word was coined was because there was growing interest in doing something truly remarkable that only comes around, every so often. A big, audacious project. The only other one in my lifetime, I can tell you, that reminded me of the Human Genome Project, which I'm going to tell you about, was the moonshot, which took place in the 1960s, where we put a person on the moon for the first time, where it was a declaration, we are going to do this, we're going to get scientists to figure out how to do this, and we're going to do it as quick as we can. And there was a drumbeat of discussion leading up to the Human Genome Project that predated the naming of the field of genomics. There was a meeting, there was a big editorial that came out that was critically important. And what I could tell you is that once the field was named, there was even growing interest even after that, through a series of reports that got put together and even congressional hearings that took place in 1989 that gave momentum to this idea that we had reached a point in time where it seemed conceivable and actually appropriate to try to read out all 3 billion letters of the human genome. And so in 1989, everything really changed forever. I think 89 was this incredibly monumental year. First of all, I should point out that 1989 is when Taylor Swift was born. I don't know about the rest of you, but I'm an admitted Swiftie. So the fact that 89 was when she was born was pretty significant. My memory of 1989 actually was not Taylor Swift being born. My memory of 1989 was that the Human Genome Project was about to be born. We were getting all organized to get involved in launching the Human Genome Project. I was at that time a trainee. I was not even yet a faculty member. I was training clinically, I was training scientifically, and I saw this as an incredible opportunity to get involved in a once-in-a-lifetime project. And so I got involved literally on day one and participated in the project for all 13 years. Now, the Human Genome Project truly was historic. It was helpful that it was successful in the end, but most importantly, it just broke the mold of most other scientific projects undertaken in biology. It was international. It was big. It wasn't one scientist. It was thousands of scientists. It was very organized, and it was very unusual in a number of ways. It approached how to do science, including the fact that it shared all of its data from the very beginning to the very end. It didn't wait till papers got published. All data was made available. And so this was truly an honor to participate in this. The project took 13 years. It accomplished all of its goals. And the signature goal among several goals was this idea of having to generate. I really want to, I don't see all of you, but I know you're a young audience. And I suspect almost for certain that the main focus of the main audience listening to me was certainly not born in 1990. So you don't know what it was like back then. So, but I know most of you are really young, and most of you are probably very technology oriented, like most youth are in America today. I want you to realize that those of us who took on the challenge of the human did this starting in 1990. We were going to do something that had never been done, and it was going to require incredible cutting edge technology, incredible abilities computationally, and incredible skills that were going to be needing all sorts of cutting edge approaches to things. What was life like in 1990? Well, let me tell you about things that did not exist at the launch of the Human Genome Project, because it'll illustrate that doing the Genome Project now would be infinitely easier than doing it in 1990. Do you realize that in 1990 we didn't have web pages? Those came out in 1991. Do you realize that in 1990 we didn't have texting? That came in 92. Do you realize that in 1990 we didn't have live streaming? We didn't have web browsers. We didn't have the Sony PlayStation. We didn't have GPS. That didn't come on until 1995. Amazon similarly became available in 95. The first Microsoft Windows came in 95, 5 years into the Genome Project. USB cables came the same year, DVDs the next year, Wi-Fi halfway through the Genome Project before we even had Wi-Fi anywhere in the world. Oh, and Google didn't even come on board. You couldn't even Google search things when the Genome Project began. Social media didn't start until the Genome Project was nearly over. None of those things were there until some point during the Genome Project. By the way, here are things that only became available after the Human Genome Project. iTunes, Facebook, YouTube, Twitter, iPhone, Siri. Imagine none of those things were available when the Genome Project was taking place. Now, you may wonder, how did you exist in 1990? It must have been prehistoric era without any of these things available when the Genome Project started. And it wasn't totally prehistoric, but we did have cameras back then. And there was a pretty good camera that got a good capture of me one day when I was coming into the lab to work on the Human Genome Project. And there you see me riding my dinosaur as I was coming out of the cave, heading to the lab to try to get the Genome Project done. Thank goodness for AI. It helps entertain people. Okay. One thing I think that is really hard for people to get their head around, especially, and I know this because I've been teaching about genomics and the Genome Project really since the beginning of the Genome Project, so 37 years, is it's really hard to get your head around what does 3 billion look like? 3 billion letters is made up of, is our genome, and every one of our cells in our body have 2 of those. They have 1 copy we got from mom, 1 copy we got from dad. I mean, it's just a huge amount of information. So there's various analogies or metaphors that can be used. So one of them is making a book of the human genome sequence. So here's what you could say, is that if you typed out the 3 billion letters of the human genome sequence with no spaces in 12-point font on 8.5 by 11 pages with standard margins, guess how many pages it would take? It would fill 1.2 million pages, and it would look like a book, something like that. 1.2 million pages. That's just remarkable. Now, another way, some of you may be computationally oriented. You may think, well, okay, that's if you actually printed it, nobody prints stuff. We store stuff electronically. I don't need to print anything out. If I go to store a human genome sequence in a computer, how much disk space do I need? Well, a person's complete genome sequence. Now, in this case, it's 2 representations of the human genome, 1 from mom, 1 from dad. So you as a person, your complete sequence, 6 billion bases, would take up about 1.5 gigabytes. Guess what? In order to operate on this laptop PowerPoint, to be able to show you this PowerPoint presentation, I actually need to use a similar amount of space, 1.8 gigabytes. So just imagine the human genome. I mean, either you think that's incredibly big or incredibly small, but the software necessary for running PowerPoint is roughly the same amount of computer space as your complete sequence would be if we put it on this computer. And so then there's one other way that I know is sometimes helpful to represent is to visually see what 3 billion looks like. So what I really want to just stress here is that the code for human requires less computer storage than the code for Microsoft PowerPoint, which, some people just think is just remarkable in conceptualizing. But as I was saying, some people find it better if you actually visualize it. So watch this video.
Hi, I'm Eric Green. I'm director of the National Human Genome Research Institute at the U.S. National Institutes of Health. We study genomes. A genome is all the DNA of an organism. It's sort of like a blueprint. Every living creature needs a blueprint to develop and then to operate. It turns out that here on Earth, our blueprint's made up of DNA. Now, it turns out that all the DNA of an organism, or in other words, a whole genome of an organism, can get pretty big. For example, it turns out that the human genome consists of 3 billion building blocks. The building blocks are called nucleotides or bases, but we'll just represent them by the first letter of their name. Turns out one building block has an A to start it, another one a G, another one a T, another one a C. So GATC is the way we represent DNA, and it's the order of the letters of DNA that encode all the information necessary to make a blueprint actually help an organism develop and operate. Well, human genome, 3 billion, that's a big number. If you go to represent it, you need 3 billion letters. Over the years, I have found that people really struggle getting their head around the idea of what is 3 billion letters that make up the human genomic blueprint. What does that look like? So here in this hallway, we put together an exhibit that helps to explain that. We basically decided to take the human genome and represent not all of it, because our hallway's not big enough, but just represent 1/1000th of it. So 1/1000th of 3 billion letters. And we couldn't make the letters very big. If we did, it wouldn't fit in the hallway. You can see we had to make the G's, A's, T's, and C's quite small. But represented here is precisely the sequence of 1/1000th of the human genome. And as you walk along, you can see all the G, A's, and T's, and C's, and their big sequence. It's actually sort of boring to look at. And that's why we represent most of this as pretty boring looking letters. Every once in a while, though, you'll see a short string of letters in another color. That's in red. What do those mean? Well, it turns out that about 1.5% of the letters of the human genome actually directly encode for proteins. Proteins are the bricks and mortars that make up our cells and tissues. Also, a group of these red sequences is sometimes called a gene. So genes make proteins. It's sort of the real workhorses of our genome. And so you can see that very few of our letters in our genome actually directly encode for protein, but those that do turn out to be pretty important, because without our genes, we really don't develop and we don't operate very well. Now, if you look at one particular gene on this particular stretch of the human genome, we highlighted that in blue to really give it a little bit of extra oomph. Turns out this is a really interesting and medically important gene. This is the gene that, when mutated in one of these red regions, causes the disease called cystic fibrosis. And so this is the famous cystic fibrosis gene. Now, we've been walking along this 0.1% of the human genome for quite some time, but we're still not done. And that's the remarkable thing about the human genome, is just how big it is. And you can see that just representing 1/1000th of it required quite a long hallway, required quite a lot of letters and quite a lot of wallpaper to sort of get up. In fact, I can tell you, if we wanted to represent the entire human genome in letters of this size, it would have required not the roughly 83 feet of this hallway. It would have required about 15.5 miles. And that we weren't going to get in a single hallway at the National Institutes of Health. That's why we chose to end it here and say that we've now represented 0.1% of the human genome. And that's as far as we can go in this particular exhibit.
So I hope that gives you an appreciation of what the genome scale is when you think about 3 billion letters. But that was the story that takes us up until 2003, the end of the Human Genome Project. And that was 22 years ago, and it was remarkable, and we celebrated, and it was an incredible human milestone. But it was as much a starting line as it was a finish line, because the truth of the matter is, finishing one major thing like the Genome Project was really just the start. And it really has been exciting over the last 22 years to see how genomics has expanded and disseminated really across all the life sciences. In fact, all of these areas you could have a keynote speaker talk about, because there's been tremendous infusion of genomics into areas like agriculture and forensics and population history and evolution and so on and so forth. These are all great storylines. It's not the storyline I'm going to follow. I was trained as a physician scientist, and for 31 years I worked at the National Institutes of Health. So it shouldn't surprise you that my focus was on applying genomics to health and disease and medicine. And particularly our institute, that was going to be what it was going to pursue in leading the US's effort into human genomics. Well, this was not a terrible surprise, to be honest with you, because there was always a recognition that a key aspect of the Human Genome Project was that it was going to give us insights about how to develop and operate a human being. And maybe those insights would be helpful in practicing medicine. because so much about the way we practice medicine is done sort of based on the average patient. We don't get very specific or very personalized or very individualized information about individual patients because we didn't have access to being able to look at their DNA in detail. But that was going to change having in hand a reference blueprint, if you will, of a human being. And so it became pretty obvious, both in the popular press and in the scientific press, that juxtaposing these two words, genomic and medicine, might be a good idea. That maybe we can use genomics to improve the practice of medicine by getting genomic information about individual people. And so this was very exciting. It was a little scary, though, because 22 years ago, we could spell genomic medicine, but it was a very blurry concept. We had no idea what it looked like, how it was going to actually be implemented, or how we were going to get there. Now, we could imagine that genomic medicine would involve using genomic information about an individual as part of their clinical care. In other words, not treating people on average, but getting some insights about their uniqueness and their DNA and using that uniqueness for prevention, for diagnosis, and for treatment. By the way, you will sometimes hear other terms like medicine, individualized medicine, precision medicine. I tend to use genomics. I'm just going to talk about the unique genomic features of individuals. These other terms tend to bring in other things like social and physical environment, and that are unique to individuals as well. I will stay on the more narrow definition of genomic medicine. So we had a pivot 22 years ago. We had just finished completing the Human Genome Project, but now we had a new journey. We had a journey where we needed to move beyond the Human Genome Project and someday implement genomic medicine, even though it was a blurry concept. Well, like any journey, it's just got to start with the first step, just like the way we did the Human Genome Project, and we can't even think we know what all the steps are. But what we do know is you do it one step at a time, and when you've conquered a step, you cross it off your list, and you say, check, 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 and eventually you'll get to the destination. I will also tell you that in the case of an audacious a journey like realizing genomic medicine, you have to realize you're going to be in it for the long haul. This is going to be a marathon. It's not going to be 13 years. It's probably going to be several generations. It's going to involve a lot of people, a lot of efforts, a lot of disciplines. And that's okay. We can handle that. We're just got to be prepared for a very long journey. Well, what has transpired? Well, if I had another hour or two to lecture with you, I could give you lots of details about what has happened over the last 22 years. But for the purpose of time, I'm going to have to be very abbreviated. Happy to answer questions during the Q&A. But there have been significant advances over the last 22 years that have inched us closer and closer, and in fact, have even allowed us to touch genomic medicine. The first of these steps related to the fact that that first genome sequence generated by the Human Genome Project, it was amazing, it was high quality, and it was expensive. It cost about $1 billion. $1 billion, that's never going to be used as a clinical test. We want a clinical test. We want a test that can be done for like $1,000 or less. In fact, for 22 years, a battle cry has been the $1,000 genome. And the idea is, can we reduce the cost of sequencing a human genome from $1 billion to less than 1,000? And the answer is we've done it. We have reduced the cost of sequencing a human genome by more than 1 million-fold. It now costs less than $1,000. It's heading towards $100. We can check this off of our list. It has been incredible, the advances in sequencing methods that allow us to routinely sequence a human genome for less than $1,000. Having done that, we of course didn't want to just have one human genome sequence, which is what the Genome Project produced, but rather we want many, many. We want to know how we're different. If you happen to be sitting in a room with somebody, compared to the person sitting to their left or the person sitting to your right, you have about 3 to 5 million letters across your genome that's different compared to the person sitting next to you. And those letter differences are actually quite common typically, and we can get information about the most common variants, as they're called, in other words, different spellings, by just sequencing a lot of genomes. And we've done that now. We've now sequenced millions of people's genomes from all across the world, and we now have developed very rich catalogs of the spelling differences that are frequent in the human population. And we can cross that off our list. We know a tremendous amount about how people vary. But of course, just because they vary in their DNA sequence, what does that mean in terms of how it influences their biology or their health? In order to understand that, you have to know, like, whether or not a spelling difference actually leads to a functional difference. Most spelling differences don't lead to functional differences, but we are increasingly understanding how the human genome functions, and we're increasingly learning how different sequences influence the function. And so we have had the human genome sequence in front of us for 22 years, and we've learned a lot about its function. We have a long way to go. Many of you hopefully will go to science and help us interpret it, and then your children will help interpret it, maybe even their children. This will be a long time to fully interpret all 3 billion letters. I'll give it a medium check mark because we've learned a lot about the human genome, but we have a long way to go. Well, with cheap ability to sequence and a better understanding about how our DNA is different from one another and how it might influence function, we've begun to understand how it is that genomic differences between any of us leads to giving us a greater propensity for a certain disease. We've greatly advanced our understanding of the genomic basis of human disease. That's been particularly the case for diseases that are relatively rare and that are caused by mutations in one gene. So these are diseases like cystic fibrosis and Huntington's disease and sickle cell anemia. And there's about 10,000 of those diseases, and we now know the genomic basis of about 6,000 of those, whereas we only used to know about 50 or 60. Now, there's a lot of other diseases that are much more complicated, including diseases that Stephen Parker studies, diseases like diabetes and lots of other common conditions where there's many, many, many genes involved, and there's a lot more influence of the physical and social environment. Those are complicated. We're making progress. But because there really is still a lot more work to do, I'm going to only give this a medium check mark. But what's really fun to talk about, and I'll only be able to just touch on, is the fact that we are now putting all of these steps together and are beginning to use genomics in medicine. So some of the earliest examples of genomic medicine are now coming forward. I'm just going to give it a little check mark because it's just the earliest examples. But it does illustrate that we are beginning to reach the point where we are realizing genomic medicine in certain clinical settings. What are those clinical settings where genomics is already having an impact? I'll just give you a handful of examples, and I won't be able to describe these in any detail, but happen to answer questions. First one is cancer. Cancer is a disease of the genome. Cells that are cancerous have a whole lot of DNA changes that are making them grow out of control. And we can use the tools of genomics to understand what the changes are in cancer cells, in many cases, better treat, better manage, develop new treatments, develop surveillance methods, et cetera, all of cancer care is getting turned upside down because of genomics in a very positive way. I think at the end of the day, this is where genomics will have its greatest impact. A second area are those rare diseases I was telling you about. Nowadays, if a patient presents, either a child or a teenager or an adult, and just don't know what's wrong with them, and a standard workup is just not yielding a diagnosis, for less than $1,000, they'll just sequence that patient's genome. And as a result of that, you will, in more times than not, figure out what's wrong with the individual. And there's certain settings where this is done very acutely. For example, it is now becoming more and more standard that in newborns who are acutely ill and are in neonatal intensive care units, these are these little children that have all sorts of, little babies that have all sorts of problems. They can't figure out what's wrong with them. They can't tell you what's wrong with them. In some cases, they will die. And now there are ways to sequence their genomes in less than 24 hours and get an immediate answer that in some cases results in saving their life. This is now becoming standard in some of the best neonatal intensive care units in the world. The number one genomic test right now is a test where pregnant individuals give a little bit of their blood, and floating around in their blood is a teeny bit of fetal DNA. And you can determine by a screening method whether there's anything wrong with the chromosomes in that unborn child. Now, this used to be done by more invasive methods like amniocentesis, which were risky, and now it could be done by a simple blood draw, such that about 7 to 8 million pregnant individuals will get this prenatal genomic screening done. It's called non-invasive prenatal genomic testing. It's the number one genomic medicine test worldwide. And the uptake has been truly remarkable. Many, many, many pregnant couples decide to get this non-invasive test done. A big word, pharmacogenomics, is just simply the notion that you can use genomics to better select the right medication for a person. In other words, why is it that there's so many medications out there for disorders like asthma and hypertension? And a lot of times the doctor will just try one, then the next one, until they get one to work. But a lot of times they don't work. We are learning more and more that if you get a DNA test done first, you could better select a medication or better select a dosage for that individual. So this is called pharmacogenomic testing. And I think in the long run, it's going to become a part of the routine use in the prescription medication. Prior to getting a prescription medication, you will get a DNA test. And the last example I always like to mention is in prevention. Increasingly, we are learning that we don't have to wait for disease, that through genomics, we could find out DNA spelling differences that maybe you learn about in childhood that tell you what kind of diet you should have so that you don't have problems with a diet that doesn't get along with you. Or in the case of adults, maybe you may be from a family that's particularly likely to get colon cancer or breast cancer. Through a DNA test, you could find it if you're really at risk. And if so, you could get screened more frequently for colon cancer and breast cancer, such that if you get it, you'll catch it early, which will lead to a greater likelihood of a good therapeutic outcome. So these are examples, and they're all good examples of where genomic medicine is now being implemented. But I can certainly tell you that this list will grow, and it'll be exciting to see what the next new areas of genomic medicine that will emerge. So what I can tell you is that we are celebrating the arrival of genomic medicine. We are certainly popping the champagne corks because it is remarkable that in 22 years after the Genome Project, we are now seeing genomic medicine in full form. But before I start to wrap up, I do want to point out that I don't want you to think that the story I've told you about the Human Genome Project, or the story I've told you about realizing genomic medicine, was a simple, straight line where there were never problems, never challenges, never moments of doubt. This illustration really tells you what research is all about. Lots of twists and turns, lots of complexities, lots of things don't work, but you just keep soldiering on, you figure it out, and you just keep going. But that is still certainly the case now, that I don't want you to think that everything is perfect in genomics, that we're getting everything we want and we don't bump up against challenges. There are so many challenges that we always must consider. And so I just want to give you some examples of what some of these challenges are that we're facing. You know, there are some big challenges that genomics faces. One of them is that I'm oversimplifying when I say that we sequence a patient's genome and we figure it all out. Because clinical understanding a patient's genome really still is quite difficult. It's not push button. You know, we can go sequence anybody's genome for less than $1,000. We could do it in a day or two if we needed to. And we can go round on that patient. But when we round on that patient and we see there are 3 to 5 million variants, we're puzzled what most of them mean. Yes, we can sift through them. We can figure some things out. We make diagnoses, but we're leaving a lot behind because we just don't understand. So I want to make sure you appreciate that there's a lot of research going on understanding a lot of nuances about different variations and different genome functional elements and so on and so forth. This is an active area of research to make this anything approaching push button. It is a very manual, difficult process, and we're not perfect at it by any means. I'm sure it'll take another 10 or 20 years to get really good at this step. That's a challenge. Another challenge is more social, another area. Genomics is becoming relevant in life, and that hasn't always been the case. And with that comes some responsibilities. You know, I sometimes joke, but I actually think it's real, that, you know, it's sort of like Thanksgiving meals, at least in the United States. There's an adult table and a kid table. You know, back then, and when I say back then, like during the Genome Project, that was me at the kids' table. I mean, that was my colleagues we were just mapping and sequencing the genome. Most of society didn't even know it was really happening. You know, we had problems, but we had problems related to the project. They didn't spill out in society. If something didn't work, okay, the project went a little slower. That was back then. But you know what? Something has changed in genomics because I no longer get to hang out at the kid table. Now, because I've touched medicine, medicine is always societally complicated. Nowadays, I have to live at the adult table, where at the adult table, I'm told, no, Eric, you can't sit at the kids' table. I don't care that they're talking about iPhones, Instagram, and Taylor Swift. You have to stay here and talk about mortgages, life insurance, taxes, politics, and genomic medicine and healthcare. Yes, the adult table is the one where you have to take responsibility for things you are doing, and that's genomic medicine, and it now touches healthcare. And so that's a very different thing for our field. For a long time, society was barely aware of genomics, but now because we've touched healthcare, society is very aware of genomics as we go to implement genomic medicine. because there are grand societal challenges with genomic medicine, like how it gets paid for, where and how it gets delivered. There's now regulatory oversight because it's a clinical test. There's now issues about health equity and trying to make sure that everybody benefits from genomic medicine, not just the wealthy. Of course, there's lots of concern, like all medical information. There's concerns about genomic privacy and making sure that we are able to retain that privacy. And then, of course, there is issues around literacy. So one of the reasons I'm giving this lecture, we need more and more people understanding the fundamentals of genomics. We need the patients to understand it. We need the current healthcare professionals to understand it, nurses, doctors, pharmacists, physicians' assistants. And then, of course, we need everybody who's in training, whether they're in high school or they're in medical school or they're in college or they're in graduate school. Everybody who's going to be involved in genomics needs to be literate and help share that literacy across society. And of course, there's many other challenges that I'm just not having time to talk about. Maybe some of them will come up in the Q&A, but we recognize that with advances in medicine, with advances in science, come challenges, and some of these are societal challenges that we must address and must take responsibility for. That is why the field, on a regular basis, about every 8 to 10 years, publishes a strategic vision. This happens to be the strategic vision that we're operating on right now. It takes on these challenges. It looks to see what are the barriers, what are the steps, what are the things that have to be overcome. And we describe this about every 8 or 10 years, and this is the most recent version. And I just thought I would share with you, because I've told you now about a history lesson. I've told you now about some current exciting things going on in genomics and genomic medicine. I thought I would push you into the future, because in our strategic vision that we published back in 2020, we actually made a series of bold predictions about where human genomics might be in 10 years. And we're at the halfway point now. So I thought I would share with you a video that we made about those bold predictions as a way of sort of describing, peering into the crystal ball of where genomics might be heading over the next 5 or 10 years.
Genomics, which is the study of all DNA in an organism called the genome, is changing the way we practice medicine, how we manage and treat some previously incurable diseases, and our relationship with society at large. Since the 1950s, double helix and DNA have become household words, standing in for the historic discovery of the 4 chemicals that give us life, commonly abbreviated as A, C, G, and T. Genomics is at an age of innovation and invention like no other, all thanks to how strings of those 4 letters encode the information for creating and operating something as complicated as a human body. Springing forward from the success of the Human Genome Project, a massive reduction in the cost of sequencing a human genome, and the dawn of the CRISPR gene editing era, genomics is leading into an even more audacious future. A future that will define our collective lives for decades to come. But what could this future look like? Here is a summary of 10 bold and fantastical predictions made by experts at the National Human Genome Research Institute and the genomics community for the next decade of human genomics. While most are unlikely to be fully attained by 2030, achieving even one or more of these by that time would have lasting impact for science, medicine, and society. Prediction 1. Sequencing and analyzing a complete human genome will become commonplace for any research laboratory. Prediction 2. The role of every gene in the human genome will be known. Prediction 3. Environmental influences on our genome will be routinely used for making predictions about health and disease. Prediction 4. Genomics will no longer use social constructs such as race in human research studies. Prediction 5. Student projects involving the study of millions of people's genome sequences will be regularly featured at school science fairs. Prediction 6. Genomic testing will become commonplace in medicine, just like a standard blood test is used now. Prediction 7. It will be readily known if a given letter difference in a person's DNA is clinically important. Prediction 8. A person's complete genome sequence will be available on their smartphone in a user-friendly form. Prediction 9. Advances from human genomics will benefit all of society and not just a few. Prediction 10. Genomic discoveries and technologies will help cure more genetic diseases than ever before. How do we get there? The National Human Genome Research Institute is committed to leading research to make such predictions a reality. Learn more about the Institute's 2020 strategic vision for the future of human genomics by visiting genome.gov/2020SV. Dive deeply into a field that continues to transform medicine and will hopefully continue to bring curiosity, purpose, and compassion for people everywhere.
So in closing, let me just point out that what I've described to you has been truly remarkable progress over the last 35 years, starting with the Human Genome Project, leading to the earliest conceptions of what genomic medicine might look like, and now 22 years later, clearly seeing genomic medicine come into focus. It is very much like weaving together a story that started with a double helical structure, bringing the Genome Project, which gave us a magnified view of the human genome, and then using the threads of genomics to apply to basic science, increasingly human genetics, beginning to see this applied in genomic medicine in the hospital, and now continuing to build this and weave together this quilt with lots of people, lots of disciplines working hard to see new advances in genomics, that making genomics mainstream in medicine. And all this has come about because we once had a very small community of scientists involved in the Human Genome Project. And now, over 35 years, we've built up this remarkable interdisciplinary community of researchers and health professionals that are moving this forward. I hope some of you get excited about this and will join us and continue to build this pyramid of people, whether you go into more basic science or more clinical work. We welcome you, we need you, and it's a great, exciting field to be involved in. So with that, I will stop and I'll take questions as long as there is time for that. I think I'll stop sharing so I can see some of you. Thank you very much for your attention.